Aruca vesicaria, commonly known as arugula, is a dense repository of complex phytochemicals with significant clinical applications that exceed its role as a culinary garnish. The biological paradox of this leaf is that ingestion alone does not guarantee therapeutic benefit. In their baseline state, the plant's active compounds are biologically inert. These formulas represent the primary dormant components of the leaf tissue: dietary nitrates and glucosinolates. Without external triggers, these molecules remain in an inactive state. Arugula functions less as a direct source of medicine and more as a delivery mechanism for high-velocity biological pathways. Achieving clinical outcomes, such as blood pressure reduction, cellular chemoprevention, and bone mineralization, requires identifying the specific physiological and mechanical catalysts that activate these compounds. Understanding nutrition requires looking past the presence of specific molecules to the precise human biological triggers that initiate their function. In 2015, the journal Hypertension published a study demonstrating that dietary nitrates provide sustained blood pressure lowering in hypertensive patients. Arugula contains exceptionally high levels of these nitrates, often surpassing beetroot. However, these plant-based nitrates cannot widen blood vessels until they undergo a specific conversion. This vascular optimization relies on a single physiological catalyst, the oral microbiome. Known as the enterosalivary circuit, this biological pathway converts inert nitrates into vasoactive compounds. Commensal bacteria in the mouth facilitate the first trigger point, stripping an oxygen atom from the nitrate molecule and converting it into nitrite. The newly formed nitrates are swallowed, surviving the stomach's acidic environment to enter the bloodstream. Once in the blood, the molecules undergo a final structural shift, becoming nitric oxide gas. Nitric oxide is a transient signaling molecule. Its primary mechanical role is to target the smooth muscles that wrap around our blood vessels. When nitric oxide signals the smooth muscle to relax, it actively widens the blood vessel. This is vasodilation. This expansion changes systemic hemodynamics, improving blood flow and oxygen delivery to muscles and organs. Increased nitric oxide bioavailability provides secondary protection by inhibiting platelet aggregation and reducing white blood cell adhesion to vascular walls, limiting inflammation. The blood pressure reductions associated with arugula are the result of a symbiotic chemical reaction dependent entirely on the bacteria present in the human mouth. Moving to cellular defense, a 2007 PubMed study detailed the mechanistic basis for the cancer risk reduction associated with cruciferous vegetables. This involves a second dormant biological pathway, the enzymatic cleavage of glucosinolates. The physical architecture of the arugula cell keeps its sulfur-containing glucosinolates completely separated from the enzyme myrosinase. Initiating this pathway requires an external mechanical catalyst, such as chewing, chopping, or blending, to rupture the rigid plant cell walls. This rupture allows the myrosinase enzyme to mix with and chemically attack the glucosinolates, cleaving the molecules. The output of this cleavage is the formation of bioactive isothiocyanates and indoles. Inside the liver, these isothiocyanates modulate phase 1 and phase 2 detoxification enzymes. This modulation helps the body neutralize potential carcinogens, preventing them from inducing cellular DNA damage. Research also suggests these compounds can induce apoptosis, programmed cell death in existing malignant cells, and inhibit the angiogenesis required for tumor growth. The chemopreventative potential of arugula is essentially a botanical defense mechanism, activated for human use only through the physical destruction of the plant's cellular structure. Regarding skeletal density, a 2024 study published in PubMed found that vitamin K supplementation significantly improves bone metabolism in aging populations. While calcium is essential, the high concentration of vitamin K in arugula is an equally critical, yet frequently inactive, variable in bone health. The third necessary catalyst for this pathway is the presence of dietary lipids. Vitamin K is fat-soluble. Without a lipid carrier, such as olive oil or avocado, the intestinal absorption of this nutrient remains poor. Once absorbed, vitamin K travels to the skeletal system to interact with a specialized protein matrix called osteocalcin. In its baseline state, osteocalcin cannot physically grip calcium ions. Vitamin K structurally modifies the osteocalcin protein, physically altering its shape to create active binding sites. This allows calcium ions to successfully bind to the bone matrix, facilitating mineralization. This mechanism also addresses the risk of unbound calcium in the blood, which can lead to the calcification of soft tissues. The synergy between vitamin K and osteocalcin directs calcium away from the arteries and kidneys and into the skeleton. These skeletal protections are entirely dependent on culinary synergy. Without a lipid catalyst, the vitamin K provided by Arugula remains biologically inaccessible. Aruca vesicaria contains a sophisticated chemical architecture, but its primary compounds remain dormant without external intervention. Activating its full potential requires 3 specific triggers: bacterial reduction for nitrates, mechanical cleavage for glucosinolates, and lipid synthesis for vitamin K absorption. These catalysts drive the clinical outcomes: improved blood pressure, enhanced cellular defense, and optimized bone density. Effective nutrition requires a shift away from the idea of passive consumption. Whole foods function as interactive chemical systems that require precise physiological catalysts to reach their therapeutic potential. Dietary optimization depends on understanding the specific biological mechanisms that transform plant matter into functional cellular medicine.